Hello, my name is Neil Donison, and I'm a PhD student from Western University in London, Ontario. Today, I'm happy to talk about one of the aims of my research thesis, which examines the role of LRRK2 in the phosphorylation of threonine 175 tau associated with ALS-CTE. Research has shown that approximately 50% of ALS-FTSD patients display the presence of pathological tau phosphorylation. Research has also shown that approximately 12 to 20% of CTE patients are also diagnosed with ALS, suggesting a link between traumatic brain injury and ALS. Our lab has shown that phosphorylation of tau at 3-adenine 175 is associated with ALS, ALS cognitive impairment, and CTE. Furthermore, our lab has described a pathway in which phosphorylation at 175 results in a cascade of cellular effects leading to further tau phosphorylation and eventually fibril formation and cell death. However, one question remains to be answered, and that is which kinase is responsible for phosphorylating tau at 3-adenine 175? And that leads me into my objective, which is to determine this kinase, which is responsible for this initial phosphorylation event. And I hypothesized that LRRK2 is upregulated following traumatic brain injury, which then directly phosphorylates 3-adenine 175 tau and leads to fibril formation. The first aim was to examine LRRK2 pathology in a rodent model of traumatic brain injury. Using immunohistochemical staining, I analyzed total LRRK pathology within the first 10 days following a single controlled cortical impact in 3-month-old Sprague-Dawley rats. And qualitatively, we can see an increase in LRRK2 pathology in our TBI rats compared to our non-injured controls. Further, this closely aligns with 3-adenine 175 pathology within these first 10 days, suggesting a link between LRRK2 and tau phosphorylation. The next step was to examine the role of LRRK2 in vitro. And so HEC293 T cells were transfected with either tau or co-transfected with LRRK and tau. And Western blot analysis showed no significant difference in the overall levels of phospho-3-adenine 175 between two groups. To further validate these results, we are going to examine the effect of 3-adenine 175's phosphorylation using a G2019S LRRK mutant, which is constitutively active. We will also examine this effect in SHSY5 cells. Lastly, if this leads to no link, we will then begin to examine other potential candidate kinases which may be involved. And this includes ERK2 and JNK1, which we have shown is upregulated within the first 10 days following traumatic brain injury. Thank you so much for